Let me go ahead here quickly. I don't know where it's. Hopefully you can see this. It's a great pleasure for me to present Shen Hoen, who obtained a BS in chemistry, physics, and mathematics from Baldwin Wallace University here in Ohio. They are in Baldwin Wallace. He did undergraduate research in the research group of Diana Barco, where he investigated the kinetics and substrate specificity of human and zebrafish construct of the enzyme peptidyl arginine deaminase type 2 in order to understand its implication in various neurodegenerative disorders. Prior to undergraduate school, Shen served 10 years in the United States Army, initially as a combat medic, and then earned the coveted Green Beret, where he continued his love for medicine and as a Special Forces medic. He had numerous deployments in direct support to the operations in Iraq and Enduring Freedom, which could be another talk for him. And his interests include balancing time between doing the school, the research, spending time with his wife and his beautiful daughter, Zoe. So with that, I would like to ask Shen to share his screen and start with the seminar.
Second here. Okay, everybody can see this?
Yep. Yes, we do.
Perfect.
We hear you.
All right, so I'd like to thank everybody for being here, and of course, thank Dr. Crespo for his great introduction. Of course, my name is Sean, and I'll talk to you about my fourth year talk. I just want to take a moment of everybody's time to remember Dr. Batiscu. And so he was a well-known and respected faculty member in the department, and unfortunately passed away last month. He immigrated from Romania to the U.S. in 1967 and earned his PhD from Case Western in 71 under Dr. Joe Zola, Nobel laureate. He took interest in promoting the use of NMR in his home country of Romania, where we see he founded the first NMR lab in his home country in '95. Of course, he has many high honors and merits that were celebrated here and abroad, and he will be greatly missed. So transitioning to today's talk, I want to talk to you about the impact that amino and carbonyl functionality has on the pyrimidine core and the consequence it has on its electronic relaxation. And so looking at our figure here, we have this single and double-stranded helical high-ordered DNA structure that we're all aware of. And over here on the left, we have our primitive chemical feedstock, molecular ancestors known as protobiotic RNA. And so this primordial soup, if you will. So how these non-living chemicals evolved into molecules capable of sustaining life and passing on genetic information that began approximately 3 to 4 billion years ago is still one of the greatest mysteries, still captivating scientists today. And so a lot of work has been done over the past century on proposing plausible atmospheric conditions that would have been present on early prebiotic Earth, different selection pressures that have been proposed that would have been enforced or imposed upon these molecular candidates during these different times of chemical evolution. And so, for example, we said here we have this higher order structure, the sugar phosphate backbone, and so glycosylation or attachment of the sugar on these ancestor candidates or even the nucleobases may have been thermodynamically unfavorable during different times or conditions. They would have had to a base pair or hydrogen bond, self-assemble in water, base stack, et cetera. And so these are all great selection pressures, but one that's often overlooked is the one of photostability, and that's a key focus in the Crespo research group. So if we think that this is a molecular supply chain or assembly line, the sun would have taken a key role, our central role as the job foreman or the boss, keeping a watchful eye on productivity and this constant chemical refinement. And so there's a lot of prevalent theories in the study of abiogenesis or the molecular origins of life. And so just touching on two today is genetics first, or RNA world hypothesis that we see here on the left. And so here, self-replicating polymers are thought to have formed first. According to this theory, RNA with this ability to store information and pass it on and perform catalytic reactions came first prior to DNA and proteins. On the other hand, we have metabolism first, or managed metabolism, or lipid first. And researchers hypothesize that metabolic networks came first before RNA and DNA, where within the managed metabolism theory, RNA could have been a molecule that helped manage those metabolic networks. So no matter which theory you subscribe to, or whether you think that maybe they were happening at the same time synergistically or simultaneously, in either case, photostability would have taken a key role during the management of these different networks. So as I stated before, tremendous amount of work has gone into in the past century proposing different starting and reaction conditions that are plausible to be able to synthesize these different precursor molecules, amino acids, ancestral coenzymes, and riboenzymes, and things of this nature. And so we see here with the famous Miller-Urey experiment in the 1950s, in a reducing environment brought on by electrical discharge or impact activity, this exogenous delivery being meteorite infall that could constantly be replenishing indigenous or non-indigenous molecules to this chemical feedstock. It was shown that synthesizing formamide as a reactive intermediate could occur. Continued work showed that formamide decomposition could lead to different reactive intermediates, where then subsequent steps were shown to synthesize these canonical nucleobases we see down here with subsequent synthesis steps with 4-amino 5-cyanoimidazole and 2,3-diamino-furanitrile. And so from this point in the 50s, a lot of work by many different researchers in different fields have shown different organic molecules, or proposed different organic and inorganic precursor molecules that formed these different building blocks, leading these functional polymers and these earliest life forms. And so no matter what starting materials you focus on and what synthetic pathway that you follow, there's a common reoccurring theme with these pathways is the pyrimidine purine core, where, as we saw with Nice's presentation, even the pyrimidine core can be thought of as a functionalized pyrimidine, where we have the imidazole ring functionalized at the C5-C6 ethylenic bond. And so, as she well stated in her presentation, she showed that C6 functionalization facilitates ultrafast internal conversion to the ground state, the ground electronic state, and frustrates that access to those long-lived singlets and triplet states. And so a systematic investigation can be applied, of course, to the canonical pyrimidines we see here, cytosine, thymine, and uracil, which have been vastly studied and shown to be incredibly photostable. We see here with ultrafast internal conversion, and that's due to the retention of that C4 position to substituent. And then when we remove the key substituent at C4, we lead to something that's more in line with something that could be photoreactive or have slow internal conversion, or even increase intersystem crossing to those long-lived singlet, triplet manifolds, respectively. And so why is this important? Well, as we see here with our canonical nucleobases, but even these other molecular ancestor molecules and organic in nature would have absorbed very strongly in this ultraviolet portion of the electromagnetic spectrum. So we see the molar efficiency of absorption by our canonical nucleobases here. But if I draw your eye to this gray spectrum, this is the solar radiance above Earth's surface. And so this is the type of solar radiance that we might expect to be incident on Earth's surface 3 to 4 billion years ago. So of course, the Earth's atmosphere now with ozone and other atmospheric gases do a great job of attenuating all this UVC and some of UVB harmful radiation, where of course remainder of UVB, UVA, and the visible beyond make it down to Earth's surface. So I just want to highlight this middle region here, where we see this overlap with our nucleobases here and the solar radiance that's on Earth's surface. And to kind of foreshadow next week's talk with my fellow researcher Luis, and where we have absorption of UVB energy that allows for photoassisted DNA damage, and he'll talk about photodimerization and the ultimately leading to skin cancer. And so again, in the spectral region that we're talking about, we're supplying a tremendous amount of energy to the system that's known to dissociate chemical bonds, yet life persists or even thrives under constant exposure to sunlight. So a common theme, as we've seen this year with fourth year talks, is the study of interaction with light with matter. So as we saw last week, this week, and of course next week. And so usually when we begin to talk about things like ultrafast internal conversion, or even more generally with photophysics or photostability, we do so with the use of a Jablonski diagram. So Naishka did a great job describing the details of a Jablonski, so I just want to recapitulate some of the key points. And so of course we have absorption of our photon, which promotes an electron to a higher excited, in this case, singlet state, where we can then non-radiatively decay to the lowest excited single state if we follow Akasha's rule, where then we have a couple options, either radiatively or non-radiatively dissipating this excess energy to the electronic ground state, or perform that spin forbidden process, exchanging spin multiplicities from singlet to triplet, where then again we can dissipate that energy radiatively or non-radiatively. The main focus I'd like you to pay attention to, though, is the opportunity to reside in these longer lived singlet and triplet states, and this has a direct correlation with increased probability of performing photochemistry. And so again, foreshadowing next week, a key design feature of the photodynamic therapeutic agents that Luis is interested in the group with the thiobases or even the selenium substituted bases is sensitizing molecular oxygen. And so being a diffusional control process, design feature of his is to have an increased triplet yield, but then also increase that longevity in that long-lived reactive triplet state to then transfer that energy to molecular oxygen. So we see some typical times associated with these electronic or these relaxation processes, excuse me. Yet we note that DNA exhibits ultra-fast characteristics as well. For example, internal conversion or ultra-fast internal conversion taking place on the order of hundreds of femtoseconds or even tens of picoseconds. And so some may be wondering, well, what's the difference? How much faster is picoseconds than microseconds or femtoseconds than nanoseconds? And so I would present to you an example that I came across when I first came to the group. And the time differences that we monitor in the CRESPO research group between femtoseconds and, let's say, microseconds or longer, if I convert my life of 35 years into seconds, it would be the difference between, in time steps, the first second I was born and the second that just passed now. And so these are tremendously different time regimes that we're monitoring, either electronic in nature or all the way to, nuclear vibrational in nature. So we can extend the idea, if you think of a Jablonski as an electronic ladder, and each rung on that ladder is a different electronic state, you could think of each one of those rungs as its own potential energy surface. And so we see an example here, as we saw with Naishka's presentation, something more in line with 2-amino purine, and we have the same Jablonski. We have absorption of UV photon, where in this example, we don't have enough excess energy to surmount this energetic barrier, leading to the point of degeneracy between these two surfaces called a conical intersection. And instead, we reside in this local minimum where we dissipate that excess energy in the form of fluorescence. Now with adenine, as we also saw with her presentation, we see here we have a very flat planar molecule, this lowest energy conformation in the ground electronic state at room temperature. And so we have absorption of a UV photon, and this takes place instantaneously. And so we've completely changed the electronic distribution of the molecule, and you can think of this as an electronic isomer of ground state adenine. And so the nuclei, of course, haven't had a chance to respond to this, and as soon as I start the stopwatch, they immediately wanted to respond to this change in electronic and vibrational energy that's been introduced to the system. So they do so by moving, and we see some out of plane deformation, right? Some puckering and twisting, specifically at the pyrimidine core, and that has completely neutralized this energetic barrier we saw before, leading to near barrierless decay, ultra-fast internally converting to that conical intersection, that access point to be able to traverse from the singlet excited state to the ground electronic state. And thereby we pass that excess energy to the vibrational modes of the ground state. Very quickly, we can dissipate that to surrounding solvent molecules, bringing us back down very fast to the electronic ground state. And so taking the idea of a two-dimensional potential energy surfaces to an n multi-dimensional potential energy surface can be quite a step. But if today we just kind of think of each one of those surfaces as a topographical map, You can see here, this is the ultimate goal with these functionalized pyrimidine and purine molecules, as we see here in the 2020 and 2015 study of purine and pyrimidine, pyrimidine and purine core respectively, is that to marry experimentally obtained results with the technique that we use in the group that I'll speak about next, and then good quality, high level of theory quantum calculations that complement one another, that allows us to be able to propose plausible relaxation mechanisms. And so how do we do that experimentally? So a key staple in the research group is broadband transient absorption spectroscopy. So very, very briefly, we start with a fundamental laser source. This is quasi-monochromatic 800 nanometer light, 4 watts of power. It pulses once every millisecond, and it lasts on the order of 100 femtoseconds. So we break that into 2 fractions. The first fraction generates our pump. And so using this OPA, we get wavelength tunability from 240 to 2400 nanometers. And so this pump is what we're going to introduce the energy into the system with. So using steady state absorbance spectroscopy and maybe even some preliminary quantum calculations, we can get an idea of where best, what state of interest we'd like to populate and investigate. So the remaining fraction continues and passes our computer controlled delay stage and generates our probe. So traditionally we would have done pump and probe, single wavelength pump and probing, but you can imagine how cumbersome it would be to tune each wavelength to gain any kind of spectral information over time. And so highlighting the use of broadband capabilities, we generate a white light continuum. With the case of calcium fluoride is 320 to 700 nanometers. So we can gain that spectral information at any given time that we're taking these, you can think of it like molecular pictures almost. And so our pump and probe meet each other back in the sample chamber, where then the probe information of interest is carried back to the computer to be further analyzed. So if we look at our sample chamber here in our cuvette, we have spatial overlap of our pump and probe, and then we control the temporal overlap or the time deviation when they show relative to one another by our computer controlled delay stage. So a similar slide that we saw with Nice's presentation, I'll just recap. I'd mentioned before that the fundamental laser pulses once every millisecond, and we make that end up being every 2, so we can perform a subtraction, which gives us our delta A. So our excited state absorbance minus our ground state absorbance yields our delta A. And so if we probe prior to the pump showing up, we just monitor the ground state population. So ground state minus ground state is 0, as we see here. And then these 2 pulses meet for each other the first time at time 0, or what we deem time 0. In that case, we take a fraction of molecules from the electronic ground state, promote them or populate them to the first excited single state in this example. And then we monitor that subpopulation, how it absorbs, in this example, 570 nanometer light. And then we can monitor over time past when the pump showed up, watch the dissipation of that excess energy. And we see our kinetic trace here. Simultaneously, we can monitor spectrally the change of absorbance, right? So again, 320 to 700 nanometers, for example. We'll take a portion of our electronic ground state species, pump them to the first excited single state for this cartoon, and then we'll monitor at different time delays how the subpopulation absorbs. So when the pump and probe overlap, we populate that subfraction of molecules to the first excited single state, and at time 0, we monitor how that species absorbs. So we get a spectral feature that's attained. And after some time dt, we have repopulation of the electronic ground state, leading us with less concentration or less molecules in that excited state. And of course, monitor how it absorbs, and we see, as expected, our signal attenuates, and so on and so forth. So all in all, we collect a tremendous amount of data at one time. And so after some pre and post processing procedures, where we can at this point begin to plot these data in 3 dimensions and really get our eyes and hands on the data and notice any spectral features of interest and begin the process of assigning them to different electronic or different relaxation processes. So we see here plotted in 3 dimensions. We have wavelength on one axis, time plotted logarithmically on another axis, which is advantageous, so we can monitor those really short time changes, as we see shown here. And then of course on the z-axis we have the change of absorbance, right? So we can see positive or negative changes of absorbances. So if we compare 2 molecules side by side, If we look here on the left, we have thymidine monophosphate, well-studied and well-characterized DNA monomer. It's been shown to be incredibly photostable. And so as expected, prior to time zero, we have zero change of absorbance. And then at time zero, very quick excitation and dissipation of that excess energy back down to zero for the remainder of the 3 nanoseconds. Now, just removal of a single atom at the position 4, in this case, an oxygen atom at position 4, completely tunes the outcome of what we obtain, and we see here. And so in this case, we populate an excited state species that persists for past 3 nanoseconds for the observable window of this particular experiment. And then we also see an increase in some emission properties of this molecule. It's important to note because while thymidine monophosphate is incredibly photostable, as demonstrated here, it's complemented by its very low emissive quantum yield on the order of 10 to the minus 4. Or in the case of 5-methyl-2-pyrimidone here, just removal of that oxygen at the C4 position gives us a quantum yield of 20 to 25%, depending on what solvent you're studying in it. So this is further interesting to highlight because, again, next week, Luce will talk about photosysted DNA damage photodimers, and one of them being 6,4 photoproduct. A moiety that's implicated in the formation of that photolesion is this 5-methyl-2-pyrimidone. So you can really monitor the formation of 6,4 photoproduct by monitoring the increased emission of this particular species over time. So again, we can slice this data up kinetically, as shown here, or spectrally, and then we can begin to plot those spectral features of interest. Lost my laser pointer apparently. Okay, there we go. So we can begin to collect those spectral features, those different spectra over time. And if I draw your eye from purple to green, we can watch bands grow in over different times, purple to green again. And then over this particular time period, again, from purple to green, we can see those decay and or shifting of those spectral features, which allows us to kind of begin to propose or assign them to different relaxation mechanisms. And so I'd like to start with talking about the pyrimidine core, an investigation that we published about last year. And so the idea with this study was, is it the pyrimidine core itself that's responsible for the intrinsic photostability of the DNA, RNA monomers, or as we suspect, is it the key functionality at those positions of different chemical substitution? So as always, we start with steady state absorbance spectroscopy. And we see here we have pyrimidine, the absorption spectra of pyrimidine under varying solvent polarities. We can already begin to note different solvatochromic effects that occur. And this is corroborated with some time-dependent dental functional theory, where if we look here on this left panel, this corresponds to the strongly allowed pi-pi star transition. And even experimentally, we can see the changes of, the change of solvent polarity leaves this transition relatively unperturbed. But on this right panel, as expected, going from solvent polarities of low polarity to high polarity, we see a hypsochromic or blue shifting of that lowest energy and pi star transition. So this is important because when choosing a wavelength to perform these laser experiments with, the molecule has to actually absorb that light. And so depending on what solvent it is in, those spectral bands can change. So we can take our transient absorption results that we see here, those different spectral features of interest plotted. We can mathematically fit that data, subject them to kinetic modeling software, which allows us to then extract spectral features, or key spectra of interest and associated lifetimes in this process called global and target analysis. And then we can couple that with our Spain collaborators' high level of theory quantum calculations that very well describe the ground and excited electronic states. We see here on this slide, computationally, our collaborators have solved for key structures on these potential energy minima on these different potential energy surfaces. And likewise, those key conical intersections of those points of degeneracy between different electronic states that allow for ease of traversing between electronic state to electronic state. So again, we can take our experimentally obtained spectra of interest from target analysis. And then using those structures on the previous slide, we can simulate a transient absorption spectra of those structures as we see here in this panel. And we could take a weighted or linear combination of them and fit the experimentally obtained data, which enables us to propose relative percentages of those species at different times. This allows us to couple the experimental and computational results that are in good agreement with one another to propose a plausible relaxation mechanism for, in this case, pyrimidine. And so it was shown with the population of long-lived singlet and triplet states that, of course, it's not the pyrimidine core itself that's responsible for the electronic relaxation or the photostability that's exhibited by the DNA and RNA monomers, but really it's the key positions of functionality.